focus is diffusion. Now what's great about this Bell Ring question is we have an image to analyze and critique so that we can understand what the question is asking us. Now using our strategies, somebody tell us what are we doing here when we have a multiple choice question? Kylie. Eliminate 2 and keep 2. Very good. So when we eliminate 2 and we keep 2, the 2 that we keep, what do we do with those 2? Feed me. Out loud.
We see which one's the best answer out of the 2.
Very good. And by doing that, we always want to connect it to the question. Excellent job. All right, so when we connect it to the question, we alleviate the process of errors, right? We don't want to make mistakes because it's very simple to make a mistake, and we don't want that to happen. So with your elimination, I want your response. Tell me which letter you would like to eliminate. Andrew, give me one. A. Who agrees with Andrew? Excellent. I agree with Andrew. Good job. A is out. Not wilted leaves. That doesn't apply. That's not diffusion. We're moving water. Right? When we have a plant wilting, we're moving water. It's moving from outside the cell. From inside the cell to outside the cell. That's water being moved. So that's not diffusion. That's actually osmosis. Good. Okay, so we can eliminate A. A is out. What other letter can we eliminate? Hunter. B, swollen tissue. Also, water moving from outside the cell to inside the cell, causing your cells to swell. But remember, the focus here is water, and that is not diffusion. That is indeed osmosis. Very good. Instead, our question is dealing with diffusion, so we need to leave the letter C and the letter D available, correct? Yes. All right, so we're going to travel to our 4 corners, so remember procedure there. And when I say our key word, you're going to travel to either letter C or letter D. Go! Okay, we'll take care of you. Thank you for telling me. Good. I like what I hear. Good. I like that word. Good word. Good word. All right, class? Yes. All right, listen to your name. I choose Josh. Come on down. Everyone else have a seat, please. All right, guys, just because we all chose the letter D, does that mean that D is best? No. What do we have to do? Justify. Good words. Clarify our answers. Be solid and know that 100% that we are accurate. So Josh is going to do that for us. So the floor is yours, son.
Well, I chose D because a bottle of air freshener is a perfect example of diffusion because it goes from a high concentration to a low concentration following the concentration gradient.
Very good. So are the molecules moving from high to low? Yes. Do we agree with that? Yes. Because inside this bottle of Febreze that it mentioned, is that a high concentration or a low concentration?
High.
It's a high concentration, right? And if I were to break it open and have you smell it, would you gag?
Yes.
Yes, because it's so highly concentrated. But once we release it and we spray it into the environment, it can spread out or diffuse, right? Or diffuse into the area so that we can move those molecules from a high concentration down to a low concentration, moving across that concentration gradient. Excellent words. Great job. Let's give them a round of applause. Excellent job, Josh. All right, guys, I need you to clear off your desks. All you're going to need is a pencil in 5, 4, 3, 2, 1, 0. All right, my young scientists, today we're going to focus on passive transport. Now, we previously learned about active transport, so when we use energy, we're going through active transport. But when we're not applying energy to a concentration gradient, we are dealing with passive transport, right? Passive means no energy. Active means with energy. So without energy, we have 2 types of passive transport that we're focusing on for today, and those 2 types are diffusion and osmosis. So diffusion is moving molecules from an area of high concentration to an area of low concentration, high to low, right? It's easy to do this, right? Think about water flowing downstream. It's going from a higher concentration to a lower concentration. It doesn't require energy, right? That's easy to do. But what if I had you jump in that same stream and go upstream? Yes, would that require energy for you? Yes. Absolutely, that'd be difficult to do and it would require a lot of energy. So that is not passive transport. Indeed, that's actually active transport. Very good. So instead, when we're dealing with passive transport, we're not requiring energy and we're moving from a high concentration to a lower concentration to do, right? And this occurs in our daily life, so we're going to connect that today. So with diffusion, we're going from high to low, and this one typically focuses on air molecules, right? Molecules that are found in the air. And then osmosis is also the same definition with one exception. The molecule that we're focusing on for osmosis is water. Very good. Got this? All right, perfect. Let's see what you can do. All right, so today we're going to focus on illustrating and demonstrating how osmosis and diffusion both occur and how these processes are connected to you and our daily lives. What's so great about science is you can connect it to what's happening in the world around you. And we're going to use some examples today to do so. Febreze is spreading out. Smell. Good. Okay, so today we're going to test you. So what I'm going to do with you today is we're going to use air freshener, and I'm going to disperse or spray this air freshener across the classroom. Now I'm going to start in a corner. Okay, I'm going to start here. So my concentration is going to begin here. Do you think the concentration is going to begin to be high or low? High. Very good. So I labeled your can, and we'll leave this up all year as a constant reminder. So this is a process of diffusion. A great example, when we spray air freshener, which we do this, right? We spray air freshener, and we're moving from an area of high concentration to low concentration. And this is a visual cue to help us remember. When molecules move from high to low, we're dealing with passive transport. It does not require energy. Right? This is simple to do. So in just a moment, we're going to test it and prove that this occurs, because we can't see this happen. I'm going to spray it into the air, and I'm going to let it diffuse across the classroom. Right? And as those molecules are diffusing across the classroom, we're going to see a response to that. So what we're going to do in just a moment, I'm going to have you put your head down, and you're going to close your eyes. And there's a reason for that, because if we close your eyes and put your head down, we can focus on our sense of smell. So as soon as you smell, this apple cinnamon Glade air freshener, as soon as you smell this, I want you to raise your hand. What's your job?
Raise your hand.
As soon as you smell it. Can it take a while to get to you? Yes. Yes, so be patient, absolutely. It can take a while because we're talking about molecules. They're tiny, right? So let that happen. Take your time. Don't be impatient with it. And because we're running a test, we need a control group. What's a control group? Do we remember?
Something that is not changed.
Very good. Something that we're not going to test. So we're not going to test Will. He's in the corner, so he's going to be a good control for this experiment. So say, hi, Will.
Hi, Will.
All right, Will's going to be our observer, and he's not being tested. So when y'all put your heads down and raise your hands, he's going to monitor you. He's going to observe, and then he's going to give us some feedback of what he gets to see, since we don't get to see it. Deal? You understand directions?
Yes, ma'am.
All right, go. Experiment starts now. All right, perfect. Heads up. All right, well, discuss. I want you to stand up and tell the class what you got to observe as the control. All right, eyes on Will.
During this experiment, I observed that starting over there where it was sprayed, people smelled it first, and as it dispersed, it diffused across the
Good.
classroom, people over here started smelling it a lot after, because it went from high concentration to low concentration, and then people were homeless.
Excellent. Way to bump up your words. Excellent job. Give Will a round of applause. Excellent job. So just by doing that simple experiment, something that we do all the time, if there's an odor in the house, we spray some air freshener, right? That proves diffusion occurs. We may not see it, but we can prove it by using our observation of smell, right? And we can apply this to other things, like perfume being sprayed. This is happening all around us. Diffusion is occurring. It's constant in motion. It's very interesting, right? Thankfully, it reaches equilibrium where we get to a point where we feel like we don't smell it anymore, right? And really, it's still there, right? The molecules are still there. It's not being created nor destroyed. Remember in 6th grade you learned that. So understand that those molecules are still in the air. They're just evenly dispersed. Once equilibrium is reached, the goal has been met. How awesome is that? All right, so we are going to apply what we're learning to a little hands-on experiment. So what I want you to do is you have roll cards on your table. I want you to set up your roles. You can identify yourself as a runner, as a leader, or as a recorder. This is for y'all. All right, class? Yes. All right, do you have your roles established? Yes. All right, perfect. Keep your roll card in front of you in case you forget your role. Now, remembering our roles, you're all going to have a part in this, so I want to hear your feedback. Your ideas and your opinions matter. So what we're going to do with our little Post-it notes and our bowl of cherries is we're going to walk through a little experiment, okay? So in front of you, you have a bowl of cherries, and there are 3 different types of cherries, and they're identified by little Post-it tags as A, B, and C. Okay? Your job is this. As a small group, I want all members, so every scientist at your table is going to collaborate your ideas and your thinking, discuss your observations. You're going to go through each type of cherry, A, B, and C, and determine maybe you see some differences. Maybe there's something you'd like to differentiate between each cherry, or maybe you see some similarities, and you're going to jot them down. So my recorder, whomever you are, you're going to write on a Post-it note, and the best way to do this is to label each Post-it note so you don't forget how you're collecting your data. So if I'm the recorder, I'm going to take 3 Post-it notes, and I'm going to label them A, B, and C. And I'm going to collaborate with my team of scientists and write our observations down. Capiche?
Bye.
Do you understand your job?
Yes.
You have just a few minutes to do so. Go! Yes, sir.
Is that a real cherry out there?
They're real. They're organic. They are biotic.
Can we pick them up and look at them?
You may. That's one of your observations.
I wonder what it smells like.
Smell.
This one has a strong.
Okay.
Like a nice third. It doesn't smell.
So do you want to say a highest smelling, or what's a better word to say?
strong?
A stronger smell. There you go. Very good. Use your words. Are those cherries biotic or abiotic?
Biotic, because they were once living.
Very good. Very good. They were once living. Good job.
What's another word?
That's a descriptive word. You can write that as your data. Now, I want you to use your advanced scientific descriptive words, okay? All right, are y'all finished? Okay, in just a second, I'm going to give you your next step.
Dark brown.
I'm pretty sure cherries
Want me to take your extras? That's a descriptive word. Use it for your data collection. Take your extras. Can you hand me your extra Post-it note pad? Thank you, guys. Awesome. All right, guys, you have 30 seconds to round up your thinking.
I think A is lighter in color. Yeah, I think A is lighter in color and more homing.
and it's a little bit smaller.
It's lighter in color.
Oh, Allie, you have 2 rolls, huh? Cute. All right, class. All right, my runners, raise your hand. Where's my runners? All right, your job is going to be this. You're going to take those 3 Post-it notes and you're going to go classify them on the board where they belong in the correct column. Capiche? Good job. Thank you. If they fall, I'll grab them. It's no big deal. All right, so as you were working together, I was monitoring you and I was reading over your shoulders your responses. And some of you are really using those higher order thinking words and thinking outside the box, and I love that way of that path of thinking. So let's classify our cherries to see if we're all on the same path when it comes to classifying different types of cherries, and then possibly we could predict what happened to these cherries. So for cherry A, give me some words that you think are descriptive words to describe cherry A. Out loud, feed me.
It's a dark red.
Darker in color, okay.
Hollow.
Hollow, what else?
It's kind of like it's the most juicy one. Like
It's the most juicy, okay.
it's like juicy from like the garden or something.
Okay, so it just came out the garden possibly?
It smells.
Like it was freshly picked? Okay, all right, not bad.
Smells weird.
It smells weird, okay.
Kind of smushy, but not as smushy as the seed.
Okay, so smush and smushiness factors, it's kind of smushy. Okay,
It's like it's not there.
I gotcha. All right, let's move on to cherry B. Now with cherry B, a lot of you use the word perfect. I kept seeing that word. So with cherry B, we noticed that as far as appearance goes and its texture, it was smooth. It wasn't wrinkled or busted or too plump or too mushy, right, if you're going to use the word mushy. And a lot of you wrote the word ripe or perfect. And I thought that was a really good observation because I agreed with that. I thought that those looked the most ripe. Those were the most appealing to me. Were they the most appealing for you? Yeah, so if I were to choose one to eat out of the bowl, I would probably choose cherry B. Do you agree with that? Okay, good. Okay. All right, so moving on to cherry C. A lot of you used the word rotten, and I saw the word wrinkled many times too. So that was good observations. So it looked as if this cherry was dehydrated, right? So it was dehydrated. It obviously shrunk in size, not shrink. It shrunk in size, and molecules of water had moved out of this cherry. It was no longer juicy and full of water contents. Instead, it was dehydrated. So remembering when it comes to plant cells, what structure, let's tie in an old unit, what structure inside the cell holds water?
The vacuole.
Vacuole, good. You can feed me, that's fine. Yes, the vacuole. So if the vacuole is dehydrated, is it going to shrink or is it going to enlarge? Shrink. So is that going to change the structure of the cherry? Yes, good connection. So that we would predict that that vacuole is dehydrated, right? That this cherry needs more water, is dehydrated. Yes, sir.
I just wanted to know what was actually wrong with cherry A.
We're about to link it, okay? So I love your thinking. Hold that thought. All right, so let's link these cherries, okay, to determine if there's something wrong with them or not, or what's going on. All right, so we're going to go through a little tutorial of how diffusion and osmosis occur. A high concentration to a low concentration. Okay, I challenge you to follow this so that you can predict what's going to happen at the end, okay? All right, so as we go through step 1, we're focusing on diffusion. High concentration to low concentration. Is this water? No, this is typically air molecules. Excellent. So inside your cherries, are there air molecules inside of them? Yes. Are there air molecules around them? Yes. So the air molecules can be focused on for high to low, and as diffusion occurs, it's going to move down that concentration gradient. And remember, passive transport without energy, right? No energy is required. As diffusion occurs inside of a cherry, we're going to see molecules move, okay? Now, unfortunately, a lot of times we forget a constant rate that occurs around us. So as those molecules are moving, they're bouncing around the cell membrane of this cherry, right? They're moving, they're in constant motion. Because we learned, obviously in 6th grade as well, we learned that molecules are in constant motion, right? Solids, liquids, and gases, they're in constant motion and they're bouncing around. However, a lot of times we forget about the constant rate of gravity. So what's going to happen to those molecules due to gravity? Right, they're being pulled down to the base of that cherry. Can we see that happening? No, but science can prove it for us, right? So if we had this molecular microscope where we could see these molecules, we would see this. We would see this occurring, where they're dropping down to the bottom. But the amazing phenomenon of diffusion is going to move these molecules from a high concentration to a lower concentration. So we're going to predict that those molecules are going to bounce around from the bottom to the top. Good. From high to low. And is it going to stay in motion? What do you think? Yes, it is. So the osmosis that's occurring in these cherries, and it has occurred in these cherries, just so you know, is very evident, okay? We have to remember, though, the structure of the cherry. A cherry can be represented very similar to a cell, right? It has cells inside of it, but we can use the whole thing as an example. And around this cherry has skin. We would let that be represented as a selectively permeable membrane, okay? Who remembers this term? Selectively permeable membrane. Does it ring a bell? So if you're selective, what does that mean? Very good. You only allow certain things to enter and certain things to exit. Excellent job. Remember when we learned this with cells, we used our classroom door as an example? Allow certain students in, certain students out. So good. All right, so selectively permeable, it's going to allow certain things in and certain things out. So when it comes to osmosis, what's the focus? Water. Can it allow water to go through easily? Yes, water is a small molecule. But what about sugar? Say it louder. It's a larger molecule. Excellent job. So sugar is a larger molecule, and you can see that in this representation. We have some white molecules that represent that glucose, right, sugar. So that sugar molecule is represented well, but notice it's having difficulty passing through the cell membrane, right? It can't get out of the cherry. That's why cherries are sweet. That's why our fruit is sweet. The sugar is trapped. It can't exit. They represent water, right? And right now, are they evenly dispersed? Yes. And that's like perfect situation, right? We want that to occur. We want equilibrium to be met. But notice what's going to happen when we have outside factors will occur. What do you see happening with these water molecules, Rebecca? The water's moving out, so higher to lower concentrations. So what's going to happen once it reaches the surface of the skin of the cherry? What do you predict will occur? Okay, so it looks like it's condensating or sweating, and then what's going to happen to that water molecule? Very Very good. It's going to evaporate, right, and go back into water vapor. Excellent job tying the water cycle. So that's exactly what occurs. Look at the next one. So those those water molecules begin to evaporate, right, and fall into the water cycle. And notice that with the cell structure of this cherry, what do we predict is going to happen when we look at this cherry? Very good. Very good. It's dehydrating the cell, right? So the vacuoles are shrinking, and we would predict for this cherry to shrink in size and look wrinkly and shrivelly. What cherry does that remind you of? C. Letter C. Excellent. So that's what's happening to letter C's cherry. You were wanting to know what was occurring in these cherries? That letter C had dehydrated. Maybe there was a drought. Maybe the farmer forgot to water these plants. Something like that could have occurred, right? So that was the end result for that. What about in another situation? If we had a lot of rain, like for example, we had rain yesterday and possibly today, it looks like it may rain as well. So if we had cherry trees out front, they would look like this, right? They'd be receiving rain. What's going to happen next?
They're going to be enlarged.
They're going to, okay, I like that you said that. He said they're going to swell. So where's the high concentration of water? Outside the cells. So if it's going to move from high to low, where's this water going to move? Into the cherry. So good. So as the rainfall hits the cherry, it's going to pass through this selectively permeable membrane, right? Very good. And then as it passes through, it's going to fill inside. And we're going to see that movement occur because the demonstration is going to show how water molecules can pass through and get inside the cherry. And once it travels into the cherry, it's going to diffuse, right? Spread out. And as they spread out, they're going to reach equilibrium, right? So we would predict for this cherry to very good, or become plump in size, right? It's going to enlarge. Diffusion is going to occur. Osmosis will occur. All those molecules will spread out. Water molecules are moving. Notice even the sugar molecules are moving. They're bumping into one another. Air molecules are moving. We have both diffusion and osmosis occurring together. How interesting is that? So this is more like letter B, right, where the cherry looked really well, right? It looked appealing. It was very ripe. However, what if it got too much rain? Yes, it's almost like it's overripe. I like that you said that, and it will burst. And if you notice in letter A for cherry A, that's kind of what happened there, right? Maybe it got too much water, too much rain, or maybe it fallen off the tree and it was in a puddle of water and it absorbed all that water, right? Took it in, it became more plump and it burst. And we'll see cracks and things of such on the cherry surface, which isn't nearly as appealing. We don't truly want that to happen, but we would predict for it to get larger and become more plump. All right. Are you able to make the connections of how this happens around you? So we could plug in any other fruit, couldn't we? Yeah, it doesn't have to be a cherry. It could be an apple. It could be grapes. It could be any other fruit. And this happens all around us. And really, we want osmosis to occur, but we want it to occur evenly. Assignment. Now, remember how before we do our science probes, we have a big idea thinking probe to do today. Okay, and I'm going to challenge your thinking to see how smart you really are. So when it comes to this activity of learning the differences between diffusion and osmosis, we're going to test that skill. I want you to prove to me that you truly understand. And by doing so, we're going to read a situation, or scenario rather, that occurred. And you and I are going to agree with either one of the students that are listed below, A, B, C, or D, like always. And also, we will be using our Plickers cards. So my Lord of the Rings people, go ahead and grab your Plickers cards. Disperse them to your table for me, please, foremost. All right, so with this thinking probe, what we're going to do is we're going to analyze the story that's given to us. We have an experimental procedure to read over. And then, as we read through this experimental procedure, we're going to identify which student we most agree with, okay? And I need you to justify your answer. So we all have test boards at our tables. I want you to set up test boards because I need individual thinking. We are going to discuss and build on this, but initially I want individual thinking. So go ahead and take a moment. Set up your test board before you answer your question. And you have a few minutes to go ahead and reread this question if you need to. Choose the best student to agree with and justify your answer. Any questions? Go.
I was wondering if the students have signed a waiver saying they can be on here.
No, we have not. But we can get them to do that today. I can make copies of my off hour and have them sign. Okay, so let's reread. Anna, I predict that all students will result in being near the center, so the center of the classroom. So she's saying that after it occurs, all the students will gather in the center. So decide whether or not you agree or disagree with that student. Good words. High five. Higher order, scenario, diffusion. You're doing great. Excellent. Very proud. All right, my scientists, you have 1 minute. All right, class? Yes. All right, let's put our pencils down. Grab your Plickers card. Now, remember, going back to your probe, you have letter choices next to each student, right? We have A, B, C, and D. So make sure that correlates to your Plickers card. You know how to follow that. So on the board, it's also here for you in case you forgot. So if you chose Leslie, I need you to choose letter A. If you chose Anna, we have letter B. If you chose David, we have letter C. And if you chose Sam, letter D. Capiche? All right, hold them up. Let's scan. All right, perfect. All right, so on the screen, we can see our results. So what are we showing, guys? What do we see? Right, so we would eliminate B and C, right? None of us agree with student for letter B. We don't agree with Anna. We do not agree with David. That's excellent. Those are the two I wanted you to throw out. So excellent job there. So your elimination process is on point. However, when we go through A and B, our best answer is actually going to be letter D. Our answer choice that was best was actually Sam. So just to clarify this, and we'll build on this more so tomorrow, or rather Monday rather, and so that we can clarify the differences between these two. But with this big idea, I want you to explore before I explain. So stack your test boards. Lower the range, you may put the Plickers cards back. For the next minute, I need you to talk to your table to justify and reason with them on why Sam is best. Go.
Can we erase all this?
No, you don't have to.
Oh, thank God.
You just have to be able to justify it. But thank you, because you're going to have to see why. Okay.
Can you explain to me why?
Then if they reach homeostasis, they won't keep on moving to where if it already reaches an equal state.
I was thinking that would have been like
Each would be evenly spread out in like the amount. You know it's waning.
Throughout the classroom.
Good. Diffusion. So if you are confused, I have a great job for you. Turn the paper over. On the back, you're going to illustrate how those molecules will move. I have a before and an after. And then also, we have a tiered activity to differentiate understanding, where you can apply increasing temperature and what would occur with those molecules, then possibly decreasing the temperatures and what would occur with those molecules. This is going to be a homework activity, okay? And on Monday, you and I are going to go over your answers. So what I need you to do is you may begin this now. That way, if you have questions, you can ask me. Okay? Do you understand your job? Yes. Capiche? Go. You may, absolutely. And I wanted to tell you, so see how this is the before picture? So this is like the lab coats. The X's are the lab coats, and the O's are the other students. So after they bump into 5 balls, they're going to randomly move, right? Those 6 X's, they're not going to stay together. They're not like a team, you know? They're not connected or bonded. So those molecules will randomly move and disperse until equilibrium is met.
Okay, but you're just supposed to put the X's anywhere in the circles?
So it's a prediction. You're right. It's absolutely it's a prediction. You're going to show me what you predict. Okay? If you're confused, read over the directions one time.
Okay, so I'm gonna start with the questions one
Okay.
time. When the temperature is increased, isn't the air thicker? Like,
In the air, the amount of water vapor in the air. So when you're increasing temperature, you're increasing energy. Does that help lead you?
so don't they move faster?
Possibly. That's a good idea. Maybe you should write that down. All right, class? Yes. All right, listen carefully. This is homework, and it is due on Monday. Also, I will send you a reminder to remind you that these are due. On Monday, we will clarify osmosis and diffusion. So make sure you have this resource with you because I need to discuss this with you. How do you feel about diffusion and osmosis? Great. Better? Okay, it'll even be better on Monday. Have a great weekend. You are dismissed. Bye, guys. Do not lose this, please.